Vegetative propagation in plants. Vegetative propagation is a form of asexual reproduction in plants. Vegetative propagation is very common in higher plants. Vegetative organs such as roots, stems, and leaves participate in this type of reproduction. Modes of vegetative propagation may be grouped under natural method and artificial method. Vegetative propagation is one way that plants reproduce asexually. Instead of reproductive parts present in flowers of a plant, some non-reproductive parts participate in this type of reproduction. This means that new plants are always made from organs that already exist in plant like root, stem, or leaves. These vegetative organs bear adventitious buds on them and are capable of producing new plants. Vegetative propagation through roots. This is sweet potato plant. These are its roots, which are modified form of adventitious roots. They become tuberous due to storage of food. These tuberous roots are also present in dahlia, asparagus, tapioca, etc. Apart from having stored food, the roots have buds also. On getting suitable environmental conditions, these buds start growing. The growing bud, when gets detached from the parent plant, gives rise to new plants, thus performing the function of vegetative propagation. Vegetative propagation through stem. The stems are efficient means of vegetative propagation. Some weak stems, when they touch the ground, give rise to adventitious roots. In certain plants, the underground stems become modified for the storage of food during the active phase of the growth and bears number of buds. In both these cases, on getting suitable conditions, the part of the stem starts growing from its bud and develops into a new plant. In chrysanthemum, the subaerial stems grow obliquely upwards and directly give rise to leaves. Such subaerial stem is known as sucker. In mint plant also, a slender lateral branch arises from the base of the main axis touching the ground. It runs for some distance and then gives rise to new plant by giving roots at the nodes. Vegetative propagation through underground stem. In some plants, underground portion of the stem gives rise to new plants. Underground stem is a modified form that serves two functions. It stores food and starts germinating on getting favorable conditions. The underground stems may be variously modified into the following kinds: rhizome, tuber, bulb, and comb. Vegetative propagation through rhizome and tuber. Rhizome is a horizontally creeping underground stem with distinct nodes and internodes. They grow sideways in the soil and have a shoot with leaves. Examples of such underground stems are ginger and turmeric. Thin, dry, brownish, papery scale leaves are present on the nodes. Axillary buds are also present on the nodes. When the conditions are favorable, the buds grow into aerial shoots to form new plants. This is turmeric. This is practiced by the farmer for growing new plant. Small plants having nodes are grown in the field. Tuber is a swollen, modified stem structure that functions as an underground storage organ. Since this is a stem, it has many buds all over its surface. These are called eyes in common language. Each one of these eyes is capable of producing into a new plant on getting suitable conditions. In fact, a whole row of potato plants can be started from just one good tuber. Tuber of Jerusalem artichoke also bears buds over it. The portions containing one or two buds are used to grow new plants. More examples of plants reproducing through underground stem. For plants such as onions, garlic, tulip, daffodil, the bulb is the unit of vegetative propagation. Bulb is a shortened underground stem to which fleshy storage leaves are attached. The axillary buds present in the nodes from where the leaves arise may develop into new bulbs in subsequent years. If you plant one daffodil bulb, a few years later, several plantlets may grow from that one bulb. They are separated and used to give rise to single plant from each bulb. There are some more plants which reproduce through underground stem. Corm is a condensed form of rhizome which is formed vertically in the ground. Corms also have a short stem, but in this case it is the stem itself which swells and stores food. The circular leaves form only papery scales. As with bulbs, the terminal bud grows into a flowering shoot and the lateral buds produce new plants. Sometimes a single corm may develop daughter corms from lateral buds, as in Colocasia, Gladiolus, Amorphophallus. Vegetative propagation through leaves. Leaves of a number of plants produce adventitious buds in their marginal notches. In Bryophyllum plant, the buds are produced along the leaf margin. These buds form roots at their base. When these plantlets break and fall from the parent leaf on the ground, a new plant is formed. Due to wind or other agents, these plants fall down in the soil, and when they receive water, light and minerals, they start growing into mature plants. This goes on and helps Bryophyllum plant to reproduce its own species without any big efforts. Besides natural methods of vegetative propagation, artificial modes of propagation are also used. Some common methods like cutting, layering, grafting and tissue culture are used by farmers, gardeners and scientists. The vegetative parts of plant body such as root, stem, leaf, etc. are used to produce new members of the plant. You must have seen gardeners taking a cutting of the stem of a rose plant and putting it in the soil. Through these artificial methods of vegetative propagation, plants having desirable or beneficial characteristics can be multiplied easily and at a faster speed. Vegetative propagation by cutting. Any portion of a plant, a piece of stem, leaf or root which has been removed from a plant with an objective of growing a new plant from it is called cutting. In rose, sugarcane, bougainvillea, etc., stem cuttings are used to produce new plants. In this process, stem cuttings with some nodes and internodes are placed in the moist soil, which gives rise to adventitious roots and a new plant subsequently. It is a very common method of vegetative propagation. Sugarcane is though grown using this method only. In rose plant, cutting is much easier to do. We just need to cut a stem from a parent plant slightly beneath the node, or a slanting cut is given to the portion having 1 or 2 nodes. Remove the leaves with a sharp knife so that the bare stem is left for planting. Reduce the upper leaf to half of their length to reduce the rate of transpiration. Place the cutting gently into the soil. After a few days, observe the adventitious roots come out from the underground node. In some cases, as in dahlia, even root cuttings can yield a new plant. In other plants like money plant, a leaf cutting may give rise to new plant. Layering. In this method, a branch which is very close to the ground is bent down and covered with moist soil, leaving the growing tip above the soil. A ring of bark is removed from the stem before it is bent down. In a few weeks' time, when enough roots have developed on the underground portion above the ringed part, it is clipped off from the parent plant and grown separately as an independent plant. This is a common practice with plants like jasmine, strawberry, etc. Roots arise from the scraped part after a few weeks. This branch is then detached from the parent plant, grows into a new plant after plantation. Layering differs from cutting, as in layering, the development of adventitious buds is induced before the stem is cut to form the new plant. Sexual reproduction in plants. Living things need to reproduce. Plants reproduce by both modes, sexual as well as asexual. The plants that reproduce sexually have the reproductive structures called the flowers. Flowers contain the reproductive structures stamens and carpels. Stamens are male reproductive organs and carpel is female reproductive organ. These two parts directly participate in sexual reproduction. Stamens produce pollen grains, while carpels contain the egg cells. A flower has four main parts: sepals, petals, stamens, and carpels. Sepals form the outermost whorl, which is often green in color. Sepals collectively form calyx. Petals are often brightly colored and collectively form corolla. They serve to attract bees, birds, etc., which are the agents of pollination. Female reproductive parts. The individual units are called the carpels or pistils. A flower may have one or many carpels, either fused or free. Collectively, all the carpels or pistils are called gynoecium. Each carpel is made up of the basal ovary, middle style, and the upper stigma. The stigma is a sticky structure that receives the pollen grains. The style is hollow and provides a passage for the male gametes to reach the female gametes, the eggs. The ovary is the chamber where there are one or more ovules attached to an axis. Ovule is the structure which gives rise to female reproductive cells and finally develops into seed after fertilization. Male reproductive part of a flower. Individual units of androecium are called the stamens, and collectively all stamens are known as androecium. Each stamen has a thread-like filament at the free end of which a lobed structure anther is attached. Anther is the part which produces pollen grains. Let us study the structure of anther. Anther is a bilobed structure, each lobe having two chambers known as microsporangia. Each microsporangium has plenty of microspore mother cells. Each microspore mother cell divides meiotically to form 4 haploid microspores, and the structure is known as tetrad. Therefore, a typical angiosperm anther is tetrasporangiate. Each cell of the tetrad transforms into a pollen grain. The pollen grain develops 2 cell walls, a resistant outer wall, the exine, and an inner wall, the intine. Its nucleus undergoes mitosis to form 2 cells, vegetative cell and generative cell. Types of flowers. There are different types of flower according to the presence of different parts in the same floral structure. A flower is referred to as complete if all 4 floral organs, sepal, petal, stamen and pistil are present in the same flower structure. An incomplete flower lacks any one or more of these parts. Begonia, rue anemone are examples of incomplete flowers. Flowers of rue anemone lack petals. Its sepals look like petals. There is another way to categorize the flowers. Flowers that contain both sexual parts, stamen and pistil, are called perfect or bisexual. Flowers like hibiscus, lily are very good examples of bisexual flowers, while others that contain either stamen or pistil only are called imperfect or unisexual flowers. Flowers of papaya plant are unisexual. They are present on separate plants. Pollination. For sexual reproduction to begin in plants, the pollen grains which produce the male gametes must be transferred to the stigma for sexual reproduction to occur in a plant. The transfer and deposition of pollen grains from an anther to a stigma is called pollination. Pollination is of two types: self-pollination and cross-pollination. If the pollens are transferred to the stigma of the same flower, it is known as self-pollination or autogamy. In this case, the pollen grains are transferred from the anther to the stigma of the same flower or different flower present on the same plant. When the pollen grains are transferred from the anther of one flower to the stigma of another flower present on a different plant of the same species, it is known as cross-pollination or allogamy. Cross-pollination or allogamy. Pollination is the transfer of pollens from the stamens to the stigma. The pollens are often carried by insects and other animals, but sometimes by wind and water too. So we can divide the agents of pollination into two categories: abiotic agents and biotic agents. Anemophily and hydrophily are the two abiotic ways through which pollens can be transferred, whereas biotic agents are insects, birds, bats, etc. Let us understand each with the help of visuals. Anemophily means pollination through air. It is the simplest form of pollination through wind. The wind-pollinated flowers show the following characteristics. This is grass. The flowers are unisexual. The pollen grains are light, smooth, dry and not easily wetted by rain and are produced in enormous quantities. Stamens are freely exposed with versatile anthers. The stigmas are large, feathery and well exposed to catch the pollen grains. Example: cereals. Hydrophily means pollination through water. It is the mode of pollination involving the transfer of pollen grains through water. This is Vallisneria. The plant is dioecious. On maturity, the male flowers get detached from the parent plant and float up and come to the surface of water. At the same time, The female flowers also rise up to the surface of water by straightening its coiled stalk. The detached male flower comes near a floating female flower and pollination takes place with the help of water current. Soon after the pollination, the long stalks of the female flower begin to coil down to bud level where the fruit ripens. Hydrophily does not occur in all aquatic plants. Many aquatic plants bear their flowers above the surface of water and are pollinated by wind or insects. Entomophily means pollination through insects. Insects visit the flowers not to do pollination but to collect nectar, edible pollen, or for shelter. When an insect visits a flower, its body gets dusted with pollen grains. When the loaded insect visits another flower, its body brushes against the stigma and by chance drops the pollen. Bees, moths, butterflies, all these act as the agents of pollination. Insect-pollinated flowers are usually large, conspicuous, and brightly colored. These flowers usually have a pleasant fragrance and sweet nectar. Their pollen grains are usually rough or sticky. Ornithophily. Pollination through birds. Birds can obtain only one staple food from flowers, and that is nectar. A bird goes to suck nectar from a flower and the pollens get stick to its body. The bird-pollinated flowers have funnel-shaped or tubular corollas which are brightly colored. The common bird pollinators are hummingbirds, sunbirds, and honey eaters. Chirotrophily. Pollination through bats. Bats bring about pollination only in the tropics. Bats are nocturnal animals and the flowers they visit are large and emit a strong odor. They move very fast and transport pollen to long distances. Bat-pollinated flowers generally produce more nectar and they also have a large number of stamens. After pollination, next step of sexual reproduction is fertilization. Fertilization is defined as the fusion of gametes to produce a new organism. In case of plants, the process leads to the creation of new seeds that grow into new plants. Only after pollination, when pollen has landed on the stigma, a chain of events takes place that ends in the development of seeds. Once the pollen grain reaches the stigma, pollen grain germinates, forming a narrow tube known as pollen tube. Dividing generative nucleus form two male gametes or sperm cells. Pollen tube grows down through the style to the ovary. The pollen tube carries two male gametes as well as vegetative cell. The vegetative cell degenerates after some time. And two male gametes enter embryo sac through micropyle. Once the pollen tube enters the embryo sac, its tip dissolves and the male gametes are liberated. One of the male gametes fuses with the egg cell present between the synergids and produces a diploid zygote. The other male gamete fuses with the diploid secondary nucleus and produces a triploid primary endosperm nucleus. This is called double fertilization. Double fertilization describes the process by which two sperm cells unite with different nuclei of the female gametophyte. There are 3 fusions that take place in the embryo sac. First, the fusion of two polar nuclei to form secondary nucleus. Second, the fusion of one male gamete with the egg. Third, the fusion of other male gamete with the secondary nucleus to form primary endosperm nucleus. Hence, the process is called triple fusion. As a result of fertilization, the following changes are observed in a flower. After fertilization, the zygote undergoes a number of mitotic divisions and forms a multicellular embryo. The product of triple fusion undergoes mitotic divisions to form a mass of cells called endosperm. Endosperm serves as a source of nutrition for the developing embryo. The antipodals and synergids disintegrate. The outer and inner integuments of ovule becomes the seed coat of the seed. With more time passing, the fertilized ovule goes on to form a seed through a series of processes. The ovule becomes seed. The ovary becomes fruit. Once the fruit forms, the rest of the flower parts die and fall away. The seed contains the embryo, which is made up of the radicle or future root and the plumule or future shoot. The supply of food for the embryo is from cotyledons until it has formed sufficient roots and leaves to obtain its own food. Each ovary contains one or more ovules. Some flowers, such as avocados, only have one ovule in their ovary, so their fruit has only one seed. Many flowers, such as kiwi fruit, have lots of ovules in their ovary, so their fruit contains many seeds. In pea plant, the ovary is a hollow cavity which contains several ovules. Each ovule gets transformed into a seed.